Hi, and welcome to the first episode of our video series on Vascular Streak Dieback, a growing concern in ornamental plants here in the United States. I'm Sophia Pinzi. I work at the Center for Integrated Pest Management at North Carolina State University, and I will be leading this video series. Each episode will feature conversation with specialists who will share their research and expertise. Together, we'll learn about this disease, its symptoms, its host plants, and management strategies. Today, our guest is Dr. John Bonkowski, who is the plant disease diagnostician at the Plant and Pest Diagnostic Laboratory at Purdue University, Indiana. Dr. Bonkowski is one of the leading experts in the study of VSD. In this session, we will explore what it is VSD, its main hosts, and possible management strategies. Thank you for being here, and let's get started.
Thank you so much, Sophia. So vascular streak dieback is a complex issue that we first were recognizing in 2019. That's when we were first getting those initial VSD samples. And we only began studying it in earnest in 2021. We're not entirely sure how long it's actually been around in the United States, but it has been observed primarily in nursery situations in woody plant stock. And the original hosts where we were finding VSD symptoms, or this problem, was in maple, redbud, and dogwood. Plants with VSD initially appear stunted or slower growing compared to healthier shrubs or trees, but eventually these leaves on one branch or a collection of branches will start to wilt, turn yellow, or eventually even develop marginal leaf scorch or this leaf necrosis. All of these symptoms are highly suggestive of vascular stress or water stress. We sometimes call it drought stress, and that's because something is affecting the water flow in the plant. When you cut into the wood of an affected branch, you actually will see these vascular streaks that are brown, gray, maybe even some other color that are very thin inside the xylem or the water conducting elements in the stem. So if you're affecting the xylem, you're affecting the ability of the plant to move water from the roots up into the plant canopy. So with some of these hosts, like dogwood, the symptoms may be a little bit more muted. They actually don't produce all that many streaks, and so it might have a general discoloration inside the wood. In some of the severely affected stems, you may eventually see dieback. So with the name VSD, we have vascular streaks that are inside those stems, and as it becomes more severe, it will die back over time. Often we end up seeing canker-causing pathogens. They'll be growing in that tissue as well, but ultimately we are finding one particular fungus in that, and I'll mention that in a second. Like I had mentioned, VSD is a very complex problem. So we often find other issues at play, not just VSD, that are affecting the plant, such as root rots or cankers, but it could also include cultural issues like excessive fertilization, excessive irrigation, so too much water, and deep planting. All of these can lead to stress to the plant and can reduce its ability to fight off disease, but ultimately we're finding more than one thing at once, and so it's a little bit difficult to point the finger at any one problem that's killing these trees, but VSD is present in each scenario. So in each of these situations where we do have VSD symptoms, we're finding one fungus present in those streaks, and that is Ceratobasidium. Ceratobasidium is a fungus that's related to a fungus known as Rhizoctonia, and that's a very common fungus that we see in nurseries, especially in herbaceous plant material. So if it sounds familiar, that's why. It ends up causing root rots and crown rots of these herbaceous plants, but we don't normally see it in vascular tissue of woody plants or even in woody plant roots all that often. So finding Ceratobasidium in the vascular system is also very strange. When we were trying to originally identify the fungus, we were using DNA sequencing, and we compared it against all the known fungi in the world. And it matched very closely with the fungus Rhizoctonia theobromea, also known as Ceratobasidium theobromea. It has different names, and the name changes over time. But it was called CT for short. So CT causes a disease called vascular streak dieback in cacao, or the plant that's used to make chocolate. And so based on the scientific literature about CT, we were able to find that it's very hard to work with in the lab, and also causes very similar symptoms to what we're finding here in the US. The more we worked with Ceratobasidium in the US, the more we were finding it's also really hard to keep in the lab, to grow it in culture, and even use it to inoculate plants. And all of this poses a really big problem when conducting research, because that means we have to have living plant material that's colonized by the fungus at hand at pretty much year round to be able to conduct these research projects. And that's not feasible and very hard to do, especially if we can't easily inoculate plants. So we have one major roadblock for research already. So far, Serratia basidium, the fungus, has been detected in 12 different states. This is primarily in nursery stock. And when it has been found in landscape situations, that's because these plants have been recently transplanted to the landscape from nurseries. So it's going to be primarily in those same species I'd mentioned, maple, redbud, and dogwood. But that's not the only plants that are affected. There are 25 different plant genera that can also be a host for Serratia basidium, and that can include maple, sorry, I actually mentioned that, magnolia, wax myrtles, so myrica, quercus, so oak, and many, many others. So it's going to be important for us to continue to do research to expand the host range to try and find out what other plants actually can be colonized by the fungus, because ultimately in the plant trade, this is a means for how it can spread around. There's not a lot of information on how it can spread in the nurseries itself, but when looking at the information concerning CT and cacao, we know that it produces wind-blown spores that can move from an infected plant to nearby healthy plants and infect the leaves there. Eventually, after it moves into the leaf, it will start to travel into the xylem and spread into the stems. Now, we don't know that's exactly what's happening with VSD in the United States, but ultimately, using this as a model, we have been trying to investigate how this might be occurring, and we're actually performing spore trapping in Tennessee. So they've done quite a bit of research with spore trapping methods in Tennessee, and we're going to try and do some of that in other states. I'm going to mention that a little bit later with our future goals. But we are able to detect Serratia basidium spores in the air, and that shows that it can move around naturally in the nursery from plant to plant. But also, we often are producing these nursery plants by propagation. So that could be clonal propagation or even putting bud chips and scion wood onto rootstocks. And if the mother plant where this material is coming from is colonized with Serratia basidium, it might be moving that fungus to the new plant that you're trying to grow. So ultimately, that's how we are increasing the total number of colonized plants. But then once those plants are sold, they can move across state lines, and might be why we're seeing it in some of the other states as well, is that it's moving in the nursery trade from one location to the next. So we're trying to determine a few different things. How can it spread in those plants? How can it spread in nature? And of course, how to manage it. We do have some information on managing VSD, but it's always important when you are managing a known disease problem is to know what to look for. So the symptoms I mentioned earlier on, make sure that you are familiar with those moving forward, and of course try to look for those symptoms in any plants as a grower when you're bringing in material from out of state, especially if you don't currently have this problem, because it's always important to get clean plant stock to try and stay clean where possible. And with that stock, it's always recommended also to put it in a quarantine area, or at least a different area from the main block of plants, to ensure that if symptoms do develop, that it may not necessarily be able to spread to all those other plants that are nearby. When you are dealing with disease problems, a healthy plant does a much better job at preventing disease on its own, or at least not being infected. It has its own plant defense mechanisms. So you want to manage for the overall health of the plants, and not necessarily for a growth habit or for speed of production to try and sell plants. Now, I understand that in the nurseries, you're trying to make money in selling these plants, but unfortunately, with excessive fertilization and trying to push for new growth, you're also making that plant a little bit weaker. With lush growth that can be easily colonized by fungi, and you could lead to disease when it is kind of stressed in that manner. So it's important to try and aim your practices to making the plant healthy, or at least preventing disease in making a healthy plant, and so that you don't end up having this general stress that can predispose to other problems. You want to also make sure that when you're planting your plants, that they're not planted too deep, because that's a very common factor when we're seeing vascular streak dieback. A lot of these plants are planted too deep, either inside the pots, if they are pot bound or growing in pots, but if they're field grown, sometimes they're a few inches too deep in the soil as well. And a lot of this can add to excessive stress to the roots, maybe even develop root rot in the absence of pathogens. So it's very important to make sure that you're planting at the appropriate depth. Currently in Tennessee, they are evaluating fungicide trials to kind of develop maybe the best fungicide program and involving a rotation of different chemicals for the management of VSD. At the moment, it is evaluating the development of VSD symptoms in these plants, but once we have inoculation protocols, we can at least evaluate how it can affect the overall growth of the fungus as it's growing out of plant material. So that stuff is ongoing. So I don't have exact products to name at the moment, but in all of those trials, one thing has been observed is that symptoms will be reduced, but it never effectively reduces them completely. And so you can't really cure the plants, as far as we know, with fungicide rotations. So we are going to have to evaluate how this might be able to be used to protect plants from new infections or new spread of the fungus within the block, so that it doesn't move around into new healthy material. Other research in Tennessee has shown that there's a larger amount of spores being detected in spore traps shortly after growers pruned red buds. So it's important that when you're pruning to remove the material and destroy it, because if you leave it in place, it is likely to provide inoculum for new infections and when the conditions are conducive. Typically, we end up seeing under heavy humidity, so high humidity, relatively warm temperatures, and low sunlight conditions when this fungus seems to sporulate. So it's important to kind of get that material out of there before there is those potential rain events or really muggy weather to try and prevent that spread of the fungus. Additional research in Tennessee has also shown that there appear to be differences in cultivar susceptibility to VSD. There are quite a few yellow and variegated cultivars, and most of these are actually highly susceptible to VSD as we understand it. They develop symptoms much faster than other purple or green foliage cultivars. And then also cultivars that have papery or thinner leaves often tend to seem more susceptible and develop symptoms faster than those that have thicker, waxier leaves. So cultivar choice and what you're going to grow may also affect how quickly this could spread within the nursery, but also may prevent disease development if you end up having something that's a little bit more tolerant or at least more resistant to the disease. We haven't actually found anything that is known to be completely resistant of the red buds, and so all of this that I'm mentioning about with the fungicide trial, the pruning, and the cultivar, it's all concerning red buds, although, like I had mentioned, there are a large number of plant species that we know of that are affected by VSD, so some of these general practices can be used to manage the issue with those plant species as well. But the big thing is when it comes to redbud, we don't know of any completely resistant cultivars at this time of Cercis canadensis, the eastern redbud. There is going to be some research looking into other species of redbud that might be able to help determine if there is tolerance or resistance that could potentially be bred into eastern redbud, but that is still ongoing as well.
Thank you for sharing all this useful information. What are the future research goals for the VSD working group?
Well, like I mentioned, with the management, we will be evaluating different plant species, especially the redbud species, Cercis species for tolerance. So Cercis canadensis is highly susceptible, but anecdotal evidence shows that Cercis chinensis is going to be tolerant or even maybe resistant to VSD. So we're going to evaluate that, as well as Cercis occidentalis, another species. We don't know their status at this point, so it'll just be exploratory to try and see if that can help introduce this to eastern redbud. For other management options, we are still looking at different fungicide trials as well as fungicide regimens to see what might be best to try and manage VSD. We're also going to hopefully evaluate in multiple states so that we can see that it works in different conditions. But then we're going to be looking at nursery practices or cultural practices to see what might best be kind of a best management practice for this problem when it comes to grower conditions. So looking at irrigation, amount and frequency, the fertilization regimes, when, how much, and so on. Pruning and sanitation practices. We already have some information that shows we should be improving our sanitation methods, but when can we prune? How often? What amount of material? That kind of stuff. And then also looking at like I said, the fungicide applications. We don't have a means of inoculating plants right now. We've tried a few different methods, and unfortunately, it's really difficult to work with this organism, especially since we can't keep it in culture for very long periods of time. So we're going to be continuing to look at inoculation protocols, including and as well as culturing the fungus and culture storage, so we can try and retain them year-round to try and help at least increase the availability of viable fungus for these research projects. The species itself also doesn't have a species name right now. We're calling it Ceratobasidium species because we don't know if it's truly Ceratobasidium theobromii. It closely matches with that and is definitely related, but we don't know if it's the same fungus. So we need to evaluate this a little bit further. So we are going to be studying the fungus with geneticists and some taxonomists to try and determine, is this a new species or is it Ceratobasidium theobromii? And then also, since we don't know if the fungus can actually spread in other states in different conditions, we know it likes really warm and humid weather. We know it doesn't like the sun, but will it spread in Indiana? So we're going to be evaluating spore trapping here in my state, and then also maybe one or two other states that have this problem already. So we can determine, is it moving around on its own, or are we only bringing it in from areas that have already had VSD as a problem?
Thank you so much for being here and sharing all this information with us. I really appreciate it.
It was my pleasure. Thank you for having me.
Thank you.